we're going to be discussing the dopamine biosynthetic pathway. And to start off, we have L-tyrosine. L-tyrosine is an amino acid that comes from the diet. So, if you want optimal dopamine, you're going to want enough L-tyrosine to be able to synthesize that dopamine because it's the substrate, it's the building blocks, the material that you need to be able to create that end product. So, tyrosine hydroxylase, which is an enzyme, and we're going to abbreviate enzyme like this, converts L-tyrosine into L-dopa. L-dopa is then converted into dopamine via L-dopa decarboxylase. And here we have our end product, dopamine. Very nice. Okay, and all of this is actually occurring in an axon terminal. But before dopamine can be released into the synaptic cleft, which is this gap between the two neurons, here we have the presynaptic neuron. And then over here we have the postsynaptic neuron. So for these two to be able to communicate and signal to one another, chemicals need to be released from this presynaptic neuron onto the postsynaptic neuron. But before we can release any chemicals, what we need to do is discuss VMAT2. VMAT2 basically funnels all this dopamine into a bubble. And we call this bubble a vesicle. And the vesicle contains a bunch of neurotransmitters. In this case, dopamine. VMAT, specifically VMAT2, is what packages this dopamine into the vesicle. It's called a vesicular monoamine transporter. Dopamine happens to be on monoamine. So it's getting packaged into this vesicle. So vesicular monoamine transporter. It's just putting all the dopamine into this vesicle. Eventually, an electrical signal will come down. We call that an action potential. And it's going to trigger these calcium receptors, their voltage-gated calcium channels, to open, which allows for the influx of calcium. So calcium is going to show up and cause this vesicle to fuse with the membrane and release its contents into the synaptic cleft, where it will bind a receptor. This will cause an effect on the postsynaptic neuron, and that is the whole purpose of dopamine. So we want to be able to facilitate this communication. Dopamine signaling is very important in the brain, so one thing that we can do is make sure that we're getting sufficient levels of L-tyrosine. Additionally, we can upregulate tyrosine hydroxylase because tyrosine hydroxylase happens to be the rate-limiting step in the dopamine biosynthetic pathway. So if you can increase tyrosine hydroxylase, you can increase the production of dopamine, which will, at times, result in increased working memory, executive function, motivation. But those aren't the best ways to be able to actually increase dopamine levels. See, when something is released from here, only about, I would say 10% actually bind to these receptors. So where's the rest of it going? Well, right here, we have something called a dopamine transporter, abbreviated as DAT. So that thing basically takes up all the dopamine back into the presynaptic axon terminal. So 90% of the dopamine is getting taken back in. So what do we do if we want to increase the amount of dopamine in the synaptic cleft? We can simply administer a dopamine reuptake inhibitor, which blocks this. This is actually how they treat ADHD. So Ritalin, or methylphenidate, is a dopamine reuptake inhibitor. Another thing that you can do if you don't have access to prescription medication is you can take an herb called Subroxy. It has a component called oroxalin A, and oroxalin A happens to be a dopamine reuptake inhibitor. While it might not be as potent as Ritalin, it is fairly potent. But Ritalin, I think, has a dopamine reuptake inhibition of 80%, while Subroxy is around 40%. So, still pretty significant. That's going to be a pretty big increase in synaptic levels of dopamine, where they'll go interact with these receptors and cause a greater effect on this postsynaptic neuron. So, this has